You can try try sharing there again now if you want, Joanne.
Okay.
Hello, everyone. You're very welcome to today's webinar. We're just checking our logistics here for a minute and then we'll be starting. Thanks for joining us.
Can you see that, Michelle?
Not yet, but I am just put on multiple sharing people, so hopefully that will work for you.
I'll stop sharing.
Yeah.
Share again.
Yeah, it's selected on multiple panelists can share simultaneously, so hopefully that is working. Oh, Jerry's saying they can see it. Sorry,
All right,
it's
great.
just me who can't. But that's okay. So as long as everyone else can see it, that's all good. Okay, right. So we'll get started then, if that's all right. So good afternoon, everyone. You're very welcome to today's CPD webinar here at the Discipline of Radiation Therapy. My name is Michelle Leach and I'm Professor in Radiation Therapy here at Trinity. And we're delighted today to be joined by our colleague, Professor Joanne Lysaght, who is Professor in Cancer Immunology and Immunotherapy, based here in Trinity's TTMI on the St. James's Hospital campus. Joanne is a cancer immunologist for the past 24 years, and she established her own cancer immunology and immunotherapy research group in 2012. And she has a significant size group of 3 postdocs, 6 PhDs and 3 MSc students. So that certainly keeps you pretty busy. And Joanne's other current roles are the Cancer Immunology Research Team co-lead at the Trinity St. James's Cancer Institute. She is also director of the MSc in Translational Oncology and president of the Irish Society for Immunology. So I think there's no one better to speak to us on this topic today about the intersection between immunotherapy and radiation therapy. So just to let you know, if you have questions for Joanne as she is presenting, if you please use the Q&A function, not the chat, the Q&A function, and we'll get those to those at the end. So with that, I will stop talking and ask you to start, Joanne, and thanks very much for joining us.
Thanks, Michelle, and thanks for the invitation. Can everyone check, or can you see the slides okay? Michelle, you might be able to.
That's what I'm getting, yeah.
Yes, great. Okay, yes. Great. Okay, thank you. So thanks again for the invitation. So as Michelle said, my name's Joanne Lysaght and I'm Professor in Cancer Immunology. So I'll be coming at this from an immunologist viewpoint. So forgive me for any mistakes I make in terms of talking about radiation therapy. So I think from an immunologist viewpoint, it's a very exciting combination combining radiotherapy with immunotherapy, because immunologists see radiotherapy as a very clean, very precise and powerful immunomodulator. And for us, it's a really obvious combination treatment to use with immunotherapy to improve response rates to that. And I think in terms of all cancer therapies, immunotherapy and radiotherapy, I think, are the ones that are evolving most rapidly, and it's a very exciting time to be in this field. So just to give a brief overview of, I suppose, the research that's going on in my research group, so you have an idea where I'm coming from. I'm very interested in the physical features of a tumor microenvironment, such as hypoxia, acidosis, nutrient deprivation, and really how did this impact our anti-tumor immune responses. And on top of that, how do the standard of care regimens that patients are receiving, such as surgery, radiotherapy, and chemotherapy, how do they also impact on the health of, I suppose, of our immune system? And this is really important in terms of if you're given an immunotherapy, you need a healthy functioning immune system to work. I'm not sure if those slides are changing. I'll keep going. If they're not, will you let me know? No, they're not changing. Okay, I think.
No, they're not moving on.
Okay,
sorry.
I'm going to stop sharing this and start again.
Yeah, maybe start again. Yeah. I could see your front slide, but then after that, got stuck for some reason.
I think, I don't know, is that moving?
Let's see now. hang on, I just have to sneak out again.
Apologies for this.
That's all right. We always have these little mishaps. No, nothing is coming up now, I'm being told reliably by the audience. Now it's started screen sharing again. Yeah, I can see a brief overview of my research.
Yeah,
Is I can see.
yeah,
great.
That's what I want. Hopefully that moves. If it doesn't, please let me know. Yeah, so as I was saying, the physical features of a tumor microenvironment as well as standard of care treatment that patients receive all have a really profound impact on the immune system's ability to kill cancer. The work that we do are focused predominantly around T cells because they're my favorite immune cells. And what we do is we look at immune checkpoint expression on these cells because it's a good indication of the health status of these T cells. So are they just effector T cells or are they exhausted T cells? And as I said, if you're going to introduce immunotherapy, you need a functioning, healthy immune system to see any type of response. On the flip side of that, we're really interested in immune checkpoint signaling in cancer cells. And this is something that's not really studied and it's not known very well, but immune checkpoint pathways in cancer cells can actually promote cancer and carcinogenesis as well. So that's quite an interesting area. So we know there's 5 main pillars of cancer therapy. You have your traditional surgery, chemotherapy, and radiotherapy. And then the more, I suppose, novel ones are your molecular targeted therapies as well as immunotherapy. And arguably the most successful immunotherapies for solid tumors are immune checkpoint inhibitors. But only about a quarter of patients are actually going to benefit clinically from immune checkpoint inhibitors. So that's leaving 3/4 of the patients who are going to be exposed to both short and long-term toxicities as a result and gaining no clinical benefit. So immunologists are very interested in combining standard of care regimens, particularly radiotherapy and chemotherapy, with immunotherapy to try and improve this 25% figure of response to immune checkpoint inhibitors. So I'm going to start with kind of an overview or a simplistic overview of the anti-tumor immune response. And I apologize for anyone who's very familiar with this. This is, I like to refer to the Disney version of an anti-tumor immune response. In an ideal world, if nothing goes wrong, this is what should happen. So just there on number 1, you see a solid tumor microenvironment. We know it's a very hostile microenvironment, and a lot of tumor cells can adapt to survive in hypoxia, in acidosis, no nutrients, a lot of buildup of waste. And those cells that mutate survive in that hostile microenvironment tend to be more aggressive and resistant to treatments. But many tumor cells can't adapt and they die, and they release antigens or peptides into the microenvironment. So number 2 then is a dendritic cell who resides in tissues in an immature state. They will go in and pick up or engulf these tumor cell particles. And when they do that, they internalize them and they start to mature and automatically start moving towards the nearest lymph node. Once in the lymph node, the dendritic cell, if matured fully, will activate or present that antigen that's processed to a T cell, either on MHC class 1 or 2 to a CD4 and a CD8 T cell. And it also provides a co-stimulatory signal, an activation signal to the T cell. If the T cell recognizes the peptide being presented to it, it will start to undergo clonal expansion. And for the next 7 to 10 days, those T cells will create an army of T cells, all recognizing that same tumor antigen. After that, then they will leave the lymph node. They'll travel back to the tumor site along chemokine gradients, so that's like their GPS, in the vasculature. They will then, once they reach the tumor site, they will exit from the vasculature, which is quite an energy-rich process in itself. They will start to engage with tumor cells. And if those tumor cells are expressing the antigen that they all recognize, then you will get engagement and you'll get killing of the tumor by the T cell. So as I said, this is the ideal situation if nothing goes wrong. But in reality, tumors have evolved evasion strategies that will interfere with absolutely every step of this cancer immunity cycle. So much so that avoiding immune destruction is a well-characterized hallmark of cancer. And there was a lovely paper at the end of last year by Galassi et al., which tried to summarize evasion strategies. And they named them the 3 C's of immune evasion: camouflage, coercion, and cytoprotection. And really what these are doing is summarizing the evasion strategies that interfere with every step of that cancer immunity cycle. And it's really important to understand the evasion strategies that tumors are using. And they'll differ from site to site, and they'll differ from patient to patient. But if you want to improve that 25% response rate to immunotherapy, you have to counteract the evasion strategies that the tumors are using. Otherwise, you'll never improve that response rate figure of 25%. So the type of immune cells that infiltrate the tumor really dictate whether you have tumor suppression or tumor promotion. And tumors are quite clever in that they actively recruit immune cells to the site that will promote tumor progression or protect the tumor from immune killing. And these will include regulatory T cells, myeloid-derived suppressor cells, M2 macrophages, but they also affect the fibroblasts in the tumor microenvironment to upregulate production of extracellular matrix protein. So it kind of produces this physical protection for the tumor as well. So along with the immune or tumor promoting immune cells present within the tumor microenvironment, the tumor cells themselves are rapidly proliferating. So they're using up all the available nutrients. And obviously the effector immune cells and T cells also need these nutrients to be able to kill the tumor cells. So they're at a disadvantage. And because they're rapidly proliferating, they need to generate energy very quickly. So they tend to change their metabolic processes. And as a result, they produce a lot of lactate, which makes the environment quite acidic. And again, this is a really detrimental impact on immune cells trying to kill the tumor cells. And obviously, hypoxia is a major feature of solid tumors. And as the tumor proliferates, it's outstripping the existing vasculature, and there's very limited oxygen availability for tumors. So hypoxia is obviously a key feature of solid tumors. And it's really important, as you know, in the response to radiation. And it's thought that about 3 times more radiation is required to kill hypoxic tumor cells compared to tumor cells in an normoxic environment. But from an immune point of view, hypoxia is really detrimental in terms of reducing the ability of natural killer cells and T cells to recognize and kill cancer cells. It impacts the dendritic cells, so that's the key cell that activates the adaptive immune response. So they don't process the tumor peptide, they don't travel back to the lymph node, and they can't activate T cells. Hypoxia also recruits immune cells like myeloid-derived suppressor cells, Tregs, but importantly M2 macrophages. So these tumor-associated macrophages are key for detecting regions of hypoxia, and after that they'll start releasing pro-angiogenic factors. And obviously the tumor wants to increase its blood supply to further tumor promotion. And also importantly, hypoxia forces tumor cells to downregulate MHC class 1, so they can't present antigen. So if they don't present tumor antigen MHC class 1, they're essentially hiding from the immune system. A cytotoxic T lymphocyte or a CD8 T cells can't recognize a cancer cell if it's not expressing MHC class 1. So that's an effective evasion strategy that almost 90% of solid tumors will use. And also hypoxia forces the upregulation of inhibitory molecules such as PD-L1. So unfortunately, there's no reliable biomarker for immunotherapy for patients who will respond or won't respond. So biologists have started to look histologically at what's going on in the tumor to decide if a patient would potentially benefit from immunotherapy or not. And you have a variation in terms of immune infiltrate. If there's no evidence of an ongoing immune response or no evidence of tumor infiltrating lymphocytes in a tumor, it's known as a cold tumor. These tumors can actively exclude immune cells and not let them in. And very often you'd see that, say, in pancreatic cancer cells where there's a high desmoplastic extracellular matrix, so the immune cells physically can't get into the tumor. So these patients are very unlikely to respond to an immunotherapy because there's nothing there for the immunotherapy to act on. On the other hand, you've got what's known as a hot tumor. So there's a lot of immune cells present within the tumor microenvironment. They mightn't be the ones you want, or they mightn't be effective, but there's something there to work on for an immunotherapy. So it's a good starting point. So how does radiation improve anti-tumor immunity? So you can see from this diagram, there is a big difference before and after radiation treatment. It does change the microenvironment significantly. So it can alter the tumor immune microenvironment by acting on the stroma and vasculature within the existing tumor, and that will allow physically immune cells to gain entry into the tumor microenvironment. Importantly, it can increase neoantigen availability, and this is what the immune system uses to distinguish a cancer cell from a tumor cell. And also, it forces the tumor cells to start expressing MHC class 1 again. So that process where hypoxia downregulates MHC class 1 expression, and most tumors will downregulate it, where radiation forces the re-expression of MHC class 1, so they're no longer invisible to cytotoxic T lymphocytes. Radiation can also increase tumor cell immunogenicity, and that's through the expression of danger-associated molecular patterns, or DAMPs, and probably the best studied would be HMGB1, calreticulin, and ATP release. Focal radiotherapy can activate the complement cascade, which is an evolutionary conserved innate inflammatory cascade. And again, that activates innate immune cells, and also the cGAS-STING pathway. So cytosolic DNA will activate this pathway and leads to type 1 interferon release again, which will activate an innate immune response. Irradiation also alters the release of chemokines. So I mentioned chemokines. So chemokines are the GPS really of the immune system. It's how they find where to go. They follow chemokine gradients. So an irradiated tumor will release a range of different immune checkpoints or chemokines, such as CXC, L9, 10, 11, 16, and 19. And they're really key for attracting natural killer cells and T cells, the cells of the immune system that will kill the tumor cells. They also, radiation can alter the adhesion molecules within a tumor. So once the T cells are moving back after they've expanded in the lymph node and they're moving back to the tumor through the vasculature, they have to actively get out of the vasculature and into the tumor tissue. And they do this through VCAM and ICAM expression, so adhesion molecules. So radiation can help these immune cells get into the tumor. And also, many people will describe the irradiated tumor as an in situ cancer vaccine. You've got the antigens that you need there. You've got a lot of inflammatory factors that are going to activate the immune system and effectively bypass the immunosuppression that you see in tumors before radiation. So the concept that you need an intact, healthy immune system to see the benefit from radiation treatment is not a new theory, and it was first put forward in the late 1970s. And a lot of simple experiments such as this one show that you do need an intact immune system to get a response to radiation. So this experiment just to show that irradiating a tumor in immunodeficient mice led to tumor progression, whereas irradiating a tumor in immune competent mice led to tumor regression. So that's just really simple experiments highlighting the key link between an intact immune response and response to radiotherapy. So even at radiation doses of less than half a gray, irradiated cells can release oxygen and nitrogen radicals. They activate innate immune systems such as macrophages and dendritic cells. And dendritic cells are really key cells in the anti-tumor immune response. And as I've mentioned, irradiation also causes the release of specific chemokines to recruit immune cells and anti-tumor immune cells particularly. And here you can see a breast tumor tissue in a mouse model prior to radiation and after 5-gray radiation after 96 hours. You can see the infiltration of these purple immune cells here, just quantified there in a graph. So you do get the significant infiltration of immune cells after radiation. And if the lymph nodes are outside the treatment field, naive T cells can be activated when they interact with the dendritic cells that have migrated back to the tumor draining lymph node, and they can acquire effector functions. So this concept of lymph node sparing or lymphocyte sparing is one that's, I think, being intensively studied now, particularly when combining with immunotherapy, because it's so important to keep the T cells and the lymphoid structures intact. So I'm just going to talk about immune checkpoints as they're going to be the focus of the immunotherapy that I'm going to talk about. So immune checkpoints can be stimulatory or inhibitory. They're mostly members of the B7 family. They are really key. The costimulatory molecules are really key when a dendritic cell is activating a T cell. So the T cell has to recognize the antigen, but it also needs a second signal from the dendritic cell in the form of co-stimulation, and they're an immune checkpoint. So the main role of immune checkpoints is immune homeostasis. So as soon as a T cell is activated, they start to upregulate immune checkpoint receptors with the view of terminating the T cell response. So as soon as a T cell is activated, it's looking for ways to switch off, because an uncontrolled T cell response can kill the host. They're very powerful cells and cause a lot of damage. Because it's such an powerful system, there is a lot of redundancy in this system, and it's all to protect the host from pathological immune activation. So generally, the immune checkpoint receptors are expressed by T cells. So immune checkpoint receptors would be PD-1, CTLA-4, whereas the ligands generally in healthy tissue are expressed by, say, a dendritic cell, such as PD-L1. However, tumor cells, another evasion strategy that tumor cells use is that they upregulate these inhibitory immune checkpoint ligands. So you can see from this diagram that there's a whole range of stimulatory and inhibitory checkpoints. As I said, you'd be familiar probably with the CTLA-4, the PD-1, the LAG-3. There's a lot of trials very actively looking at new immune checkpoints such as TIGIT and TIM3 as well, for the next approved immune checkpoint inhibitor. So how do immune checkpoints work? It's a very simple concept. So once a T cell engages a target tumor cell, it recognizes its antigen being presented to it. PD-1, the receptor on the T cell, will engage PD-L1 on the tumor cell. And that sends a negative signal to the T cell, and the T cell is switched off. So what immune checkpoint inhibitors do essentially is just block that binding of the immune checkpoint receptor with its ligand. Therefore, the T cell does not get switched off, it stays on, and can continue to attack and kill cancer cells. So it is a very simple concept, but that's exactly how all immune checkpoints work. But it is important to note that immune checkpoint inhibitors will only work on activated T cells. They do not activate naive T cells. They only reinvigorate an ongoing antigen-specific T cell response. So it's a common misconception, I think, that a lot of people have, in that they think immune checkpoint inhibitors can initiate an immune response. They can't. Also, immune checkpoint inhibitors will not work on a T cell if it's terminally exhausted. So if that T cell has been killing tumor cells for too long, it's undergoing a program of exhaustion towards death, and immune checkpoint inhibitors will not work on those cells. So it's a matter of getting that sweet spot of just newly activated T cells or T cells that are just slightly exhausted. So in terms of immune checkpoint inhibitors that have been approved for clinical use, obviously multiple different companies and different regions of for approvals have approved lots of different immune checkpoints, but really there's only 4 immune checkpoints that can be targeted, and that's PD-1, it's ligand PD-L1, CTLA-4, and in March in 2022, LAG-3 was approved. So it's only 4 targets. So the idea of combining an immune checkpoint inhibitor with radiation completely makes sense to an immunologist, because radiation has the ability to convert a cold tumor into a hot tumor, and all the other positive factors that radiation has in terms of altering the tumor microenvironment. And combining that with an immune boosting immune checkpoint inhibitor, for example, it makes sense that you get increased tumor cell killing. So really that concept and the hypothesis has formed the basis for a lot of clinical trials. And there's a long history, I suppose, connecting immunotherapy and radiotherapy, relatively long. But it goes back to the very first immunotherapy by William Coley, so Coley's toxins, in 1893. And this was followed by the discovery of X-ray in 1895. So both treatments have been around for a very long time. And it was really in 2012, I suppose, the last 10 to 15 years, where the combination treatments or connection between the immune response and radiotherapy has really come to the fore. So in 2012, there was the first case report of an abscopal effect in a patient. 2015 was the first proof of principle trial that combined radiotherapy with immunotherapy. 2016 started to get a bit more sophisticated, I suppose, combining immunotherapy with stereotactic ablative radiotherapy. 2017, then you had the seminal Pacific trial, and that really kind of increased the number of trials combining radiotherapy and immunotherapy. In 2019, then you had the Pembro-RT trial, which saw a doubling of the objective response rates, combining an anti-PD-1 monoclonal antibody after SBRT in patients with non-small cell lung cancer. 2020 then was the first confirmed statistic differences of the abscopal effect and increased benefit by combining radiotherapy and immunotherapy. And this finding combined two trials. And I think that's a limitation of a lot of these trials, is that they're very small. And for proper powered analysis, you're going to have to combine data. So this finding combined the Pembro-RT trial and the MD-ACC trial. And then I suppose 2021 started to use radiotherapy really as an immune modulator to reverse immune desertification. So really converting those cold tumors into hot tumors. And since then, as of yesterday, there's just under 500 trials that are active or recruiting for combining immunotherapy with radiotherapy. So it is a very active field. So what have we seen in the lab that suggests that radiotherapy and immunotherapy could improve response rates to immune checkpoint inhibitors? I'm just going to show some of the data that we have acquired over the last few years. So the cancer type that I work on most is esophageal adenocarcinoma. It's an obesity-associated cancer with an aggressive phenotype and a poor prognosis. And the 5-year survival rate is about 22% present. Only 3 out of 10 patients will respond to chemoradiation treatment, and only 1 in 4 will respond to immune checkpoint inhibition. So there's a clear unmet clinical need to identify new therapeutic strategies to improve treatment for OAC patients. So the standard of care for esophageal adenocarcinoma patients, 2 main arms: perioperative chemotherapy in the form of the FLOT regimen, and then neoadjuvant chemoradiation in the form of CROSS. So again, only 30% of patients will respond, and of those 30% of patients that do respond, 60% will develop recurrence and succumb within 5 years. The FLOT chemotherapy regimen is really toxic, and a study in our department found that only 35% of patients actually managed to receive adjuvant FLOT chemotherapy due to toxicities. So better treatment options are really needed for OAC patients. And I might be a bit biased, but I think that immunotherapy and radiation treatment have a huge role to play here. So this is just some data from Noel Donlan, who was a PhD student of mine, who's now a consultant upper GI surgeon. He's in Amsterdam at the moment, but what he did was he irradiated T cells from OAC patients with either bolus doses of 2 and 10 gray or fractionated doses of 3x2, 3x4, 3x8 gray, and looked at how this affected the T cells in terms of immune checkpoint expression, but also cytokine production. And this was all done by flow cytometry. And you can see that the fractionated or hyperfractionated doses were the best at upregulating cytokines in T cells, but also immune checkpoints as well. So it does suggest that hyperfractionation is probably the better approach at enhancing the function of T cells in these patients. And we also then want to see what effect different fractionation doses or dosing schedules would have on the tumor cells themselves. So we were lucky that we have an in-house developed isogenic model of radioresistance. So the OE33Ps are an OAC cell line, and the Ps are the parental radiosensitive, and the OE33Rs are the radioresistant cell line. And again, what we just did was bolus dosing versus hyperfractionated dosing, or just fractionated dosing, and we looked at PD-L1 expression and PD-L2. So as I'd mentioned, the tumor cells tend to express the ligands that will then bind PD-1 on the T cells. So just comparing, if you look at the white bars across the top, these are the parental cell lines, you can see as you increase the dosing, the cumulative dose, you get upregulation of these inhibitory markers, such PD-L1 and PD-L2. And it's the same, although somewhat slower for the radioresistant cell line. What's really important to note is that a decrease in expression of immune checkpoints is not necessarily what you're looking for. We found that you need at least 20% of your population to be expressing the immune checkpoint that you're going to target for the inhibitor to be effective. So if you have a treatment that's going to decrease the expression of an immune checkpoint, there's no point in giving an immune checkpoint inhibitor because there's nothing there for it to target. So obviously hypoxia is a key feature of the tumor microenvironment, and we looked at many different features, but in the interest of time, I'll just talk about hypoxia here. So again, we looked at PD-L1 expression in the parental and the radioresistant cell line, and again, you can see fractionated or hyperfractionated dosing in normoxic conditions upregulates PD-L1, both on the parental radiosensitive and the radioresistant cell line. What's interesting is under hypoxia, severe hypoxia, 0.5%, in the radioresistant cell lines, you get upregulation of PD-L1 as well. So I've mentioned DAMPs as well. These are key markers that enhance the immunogenicity of cancer cells following radiation. And this is key if you're going to combine with immunotherapy as well, because you need the immune cells to be activated and therefore be able to identify and kill the tumor cells. So again, we just did bolus dosing and fractionated dosing and looked at the effects on the parental and resistant cell lines. And you can see, again, as you increase the cumulative dose, you're increasing the expression of these DAMPs, so making them more amenable to combination or with immunotherapy as well. What was interesting is that in the parental cell line, fractionated dosing worked best in the parental cell line at upregulating HMGB1. So it does suggest that there is a differential effect of DAMP expression in radiosensitive and radioresistant cell lines. What was really interesting for us is when we started to combine radiation with immune checkpoint inhibitors and looked at tumor cell viability. And what was interesting was that the immune checkpoint inhibitors alone could reduce viability of the cancer cells. And this goes back to that area that I talked about at the start, that we're very interested in immune checkpoint signaling in cancer cells. So what is important for this talk is that when we start combining radiation with immune checkpoint inhibitors, you saw a stepwise decrease in viability, both of the radiosensitive cells and the radioresistant cells. And this was in combination with nivolumab or atezolizumab and nivolumab, so targeting PD-1 and its ligand PD-L1. What was really interesting for us was you saw in the radioresistant cell line with foregoing radiation and an immune checkpoint inhibitor combination, significantly more cell death in the radioresistant cell line compared to the radiosensitive cell line. So this suggests that maybe this combination would have an important role in treatment-resistant disease as well. So that was really encouraging. The last piece of research that I'm going to show for this talk was something that we didn't expect and we weren't really looking for. It was just one of these convenient or happy outcomes, I suppose. What we noticed when we irradiated OAC tumor biopsies from patients, that in half our cohort, we noticed an upregulation of immune checkpoints, the higher the dose of radiation, and in the other half, we saw a decrease. And so we want to know, is there anything clinically different between the patients who had an increase in immune checkpoint expression versus those that didn't? And what we found was the patients who were increasing immune checkpoints had more advanced disease, they had poor treatment response based on TRG, and they had worse biological features such as perineural and lymphovascular invasion. So this was in the absence of immune checkpoint inhibition. It was just radiation alone. So it suggests that maybe the expression of immune checkpoints could potentially be a biomarker of outcome in response to radiation. So what do clinical trials tell us about combining radiotherapy and immune checkpoint inhibitors? As I said, there's just under 500 active trials combining immunotherapy and radiation treatment as of yesterday. And while some of the trials have yielded positive results, others have been quite disappointing. And there's been a number of reasons put forward for this. One being the focus and trial design vary significantly, so it's hard to compare between trials. Patient selection is another issue. There's a lot of factors that will influence the ability or the health of an immune system, such as age, biological sex, lifestyle factors. And obviously, they're not really taken into consideration in a lot of these trials. The heterogeneity as well between patients and within tumors. A lot of the trials are very small in terms of cohort size, and therefore we need to be able to combine trial data to get any meaningful, powerful analysis as well. And therefore a lot of work is to be done, I suppose, in the trial design that they can be combined. And importantly, prior to designing clinical trials, there is a lack of complex or appropriate preclinical models that will direct trial design as well. So I'm just going to highlight a few clinical trials. I'm sure a lot of you are aware of them already. So the first one is the Pacific trial, which was a phase 3 trial for stage 3 non-small cell lung cancer patients. And it involved the administration of an anti-PD-L1 antibody after CRT. And they found that this significantly increased progression-free survival and overall survival rates compared with placebo. And it was this trial that really kind of led the way in combination trials between radiotherapy and immune checkpoint inhibitor, particularly targeting the PD-1 pathway. What's important to note for this trial is that radiation induces PD-L1 upregulation, and that can enhance the tumor cell responsiveness to an anti-PD-L1 inhibitor. And therefore, anti-PD-L1 therapies are potentially more effective after radiation. This led on to the Pacific 2 trial, which is a phase 3 trial, and again included stage 3 non-small cell lung cancer patients, but the scheduling was different. So they gave an anti-PD-L1 antibody at the same time as CRT, and this is compared to patients receiving CRT alone. In 2023, it was announced that the trial did not meet its primary endpoint of progression-free survival. And they did note an increased rate of infection in patients receiving concurrent anti-PD-L1 with CRT. And one explanation for this is that the delivery of conventional radiotherapy with immune checkpoint inhibitors caused significant T cell death, and that included the T cells that you're trying to target with your immune checkpoint inhibitor. And it's thought that trials with concurrent immune checkpoint inhibitors, but stereotactic ablative radiotherapy may be more successful because it's potentially sparing draining lymph nodes or the lymphocytes which are needed as the targets for immune checkpoint inhibitors. And then the Stellar 2 trial, which is still ongoing. So this was a preoperative short-course radiotherapy, so 5x5 gray, followed by chemotherapy and PD-1 for local advanced rectal cancer. The initial results found that 45.5% of patients in the immunotherapy group had a complete response compared to 25% in the standard treatment group. And this was a watch and wait type trial. So that was quite successful and promising. And really, the Pembro-RT trial as well in 2019. So this is a randomized phase 2 trial, again with non-small cell lung cancer, and that they found giving pembrolizumab alone or pembrolizumab after stereotactic body radiotherapy, and the dose was 3x8 gray, that they found the overall response rate at 12 weeks was 18% in the control versus 36% in the immunotherapy experimental arm. Really what's coming out of these trials is if an immune checkpoint inhibitor is given after radiation, you've better outcomes. If it was given at the same time, depending on the radiation delivery or choice of radiation, it wasn't as successful. So then the NEO-CheckRate trial is a phase 2 trial investigating preoperative immune modulatory stereotactic radiation. Again, 3x8 gray in combination chemotherapy with or without an anti-PD-1, PD-L1 therapy, and also the anti-CD73, which is an ectoenzyme that leads to adenosine accumulation in luminal B breast cancer patients. And what's interesting about this trial is that they are sparing the tumor draining lymph nodes irrespective of nodal positivity. So even if they know the nodes are positive, they're not irradiating them. And the mean dose to all tumor draining lymph nodes was less than 1 gray, and the mean dose to involved lymph nodes, if present, was 0.3 gray. So it'll be interesting to see the outcomes of that trial. But there's very few trials assessing immunotherapy alone or in combination with radiotherapy in the neoadjuvant setting. So there's still a lot of questions and unknown research to be done on the scheduling of radiotherapy and immunotherapy. So immunotherapy can be given prior to radiation treatment, and one example of this was the NRG Oncology GY017 trial. This is a phase 1 trial with 36 patients. And really they want to assess toxicity, and they found that it was tolerable, and you did get the initiation of a host immune response. And they deciphered that looking at the T cell clones and how diverse the T cell receptors were, and they found that there was an expanded T cell population after treatment. You can give immune checkpoint inhibitors with radiation at the same time, and an example of this would be the Keychain trial, which was only 8 patients. It is progression to the Keychain 2 trial. And what they found was you did get an increase in tumor antigen and activation of immune cells and recruitment of immune cells based on chemotaxis. And I suppose the most successful schedule to date is when immune checkpoint inhibitors are given directly after radiation. And a good example of this would be the Pacific 1 trial. So again, other key considerations is the delivery of radiation and what's best to combine with immunotherapy. Based on our data, we think hyperfractionated dosing is best. Ablative dosing or high dosing, anything over 12 gray, has a negative impact on the immune system. It causes lymphopenia, and also all of those immunogenic pathways that are activated, anti-tumor pathways that are activated as a result of radiation are kind of bypassed as a result, such as the CGAS-STING pathway as well. So while I've kind of highlighted the positive effects of radiation treatment on combining with immunotherapy, some of the trials, didn't give the outcomes that would be expected. And that's because there is some negative side effects of radiation treatment on the immune system. And just to recap, the positives for radiation is increased neoantigen expression, activation of the immune system through DAMP expression, activation of inflammatory cytokine pathways such as CGAS-STING, and the recruitment of immune cells. However, the negative effects of radiation treatment on anti-tumor immunity can include increased expression of immune inhibitory signals such as PD-L1, the expression of ectoenzymes such as CD73, which leads to an increase in extracellular adenosine, which is potently immunosuppressive. You have the activation of TGF-beta family members, an increase in TGF-beta release, which is a potent immunosuppressive cytokine, and that will impact every step of the cancer immunity cycle. Also, a newer member called activin A, which causes the conversion of a T helper T cell to a regulatory T cell. And then you have the release of chemokines that recruit immune cells such as myeloid-derived suppressor cells, regulatory cells that are going to help the tumor grow. So radiotherapy can expose healthy tissue to some radiation, leading to activation of the innate immune system and inflammation in healthy tissue. And immune cells are among the most highly radiosensitive cells in the body. An example of T cells are very sensitive to doses above 0.5 gray, and they're particularly sensitive when they're in the lymphoid tissue, such as lymph nodes, where they're rapidly dividing and they're proliferating, undergoing clonal expansion. And so they're more vulnerable when they're in the draining lymph node or the lymphatics than they are in the tumor microenvironment, because they're differentiated once they reach the tumor microenvironment, so they're not proliferating at that stage. Also, radiation can increase fibrosis within the tumor microenvironment, and that helps exclude immune cells physically from the tumor microenvironment as well. So lastly, I'm just going to talk about the abscopal effect. As you know, it was described by Moll in 1953, and clinical reports in humans are rare. And it is defined as a systemic immune response mediated by the effects of radiation on the immune system. And this paper just came out a few weeks ago, and I just thought it'd be interesting for this audience as a way to explain the abscopal effect, because the biological mechanism behind it is still relatively unknown. But here, this group, well, there were 60 plus authors on this paper. It is a heavy paper, but it's a nice read. And what they found was incidental or low dose radiation to the intestines synergized with anti-PD-L1 to eradicate non-irradiated tumors. And the abscopal effect of intestinal low dose radiation depended on the dose. So it only worked between 1 and 3 gray, and also tumor volume was a consideration. And what they found was the gut microbiome governs the abscopal effect of this low dose radiation of the intestines when combined with anti-PD-L1 immunotherapy. And they found certain bacteria species such as C. minuta caused the movement of dendritic cells from the mesentery to tumor draining lymph nodes where they activated T cells. And it was those T cells then that carried out the abscopal effect on non-irradiated tumors. So if patients had a certain prevalence of certain microbiome or bacterial species, it dictated whether they had an absorptive effect or not. So that was just quite an interesting paper. So I'll just leave you with persisting challenges for radiotherapy with immunotherapy combinations, and we've discussed dose and scheduling. Obviously, I think lymphocyte sparing in the blood, in the lymphatics and the tumor draining lymph node is something that's going to need a lot more research, especially when it's going to be combined with immunotherapy. Predictive biomarkers, again, identifying patients who potentially would respond to this combination treatment, and potentially gut microbiome is a way of identifying these patients. And then obviously the type of radiation that's delivered. So other considerations in terms of patient selection that impact the immune response are age, biological sex, obviously tumor volume, lifestyle factors, including obesity as well, has a big impact on the immune system. And then chemotherapy. Most patients are going to be receiving chemotherapy as well, and that has a profound impact and lasting impact on the immune system. The modality of radiotherapy, and the audience here will be far more familiar with all of these than I am, but I find what's really interesting is that spatially fractionated radiotherapy that could potentially have really beneficial effects when combined with radiotherapy. And obviously combining other immunotherapies, I focused on a few immune checkpoints, but there's newer immune checkpoints that are being examined as well in clinical trials. Also, CAR T cells are being designed with a radioactive payload. Then combining HDAC inhibitors or PARP inhibitors as well with radiation and with immunotherapy. Senolytics, again, to kill off any senescent cells in the tumor microenvironment, and then looking at STING agonists as well. So there's lots of potential combinations that could be looked at going forward. So again, it is a very exciting space to be in. And with that, I will finish up, and thank you for your attention.
Thanks a million, Joanne. That was extremely interesting, and you made a really highly complex topic very accessible. So thanks very much for doing that. I have like literally 2 pages of questions here myself for you, but seeing as how I can hop downstairs and
we can have a chat.
We will go to the Q&A. So thanks everyone for putting your questions in the Q&A. So I will start with, there are many checkpoints. Is it surprising that targeting just one, example PD-L1, PD-1, can have such a big effect in the clinic?
Yeah, I suppose this goes back to, do you consider that a big effect? There is incremental improvements by targeting one. There's a lot of approvals now coming through combining multiple checkpoints or targeting multiple checkpoints. So PD-1 and CTLA-4, the two main immune checkpoints that have been approved, and they have, you know, more benefits than a monotherapy. It really, I think, again, there's room for 75% of patients to respond better. And I think looking at there potentially be tumor-specific immune checkpoints. So a certain tumor type might have another immune checkpoint that might be more appropriate to target. Like we look at, we're finding TIGIT is higher in females than males, and maybe sex has something to do with in terms of targeting immune checkpoints as well. I think it's still really in its infancy, and there's so much that can be done to improve response rates. But there is ultimately, like the patients do, if they respond really well at the start, they tend to continue responding, but some patients develop resistance to it, and that comes back to the redundancy in the system, where their immune checkpoints are taken over.
Okay, thank you for that. I'll go, just to be fair, I'll take, because some people have multiple questions, so I'll just take one from every person first. So regarding safety and efficacy, does combining radiotherapy and immunotherapy increase an individual's risk of radiation toxicity? Probably toxicity.
Yeah, toxicity. I suppose I can talk better. Yeah, from the immune toxicity as well. Generally, when you're combining any treatments, you're not knocking out toxicity from one. You are, the patient's any combination treatment, you're going to increase toxicity. So I'd imagine the same when you're combining radiation with immunotherapy. Yeah, they're quite different toxicities.
Yeah, I mean, I was kind of wondering that myself about, with some of the trials that you showed that kind of ended early, was toxicity a factor in it? I know one, like you said, didn't meet its primary endpoint, I think, of progression-free survival. But I was wondering about is the toxicity, I'm thinking particularly of the patients who are on chemo, RT, and immunotherapy. I mean, that has to be a very toxic treatment. But I think that's probably more a question for clinicians than it is for us, probably, as is maybe this one. Sorry for the very, very basic question. No question is basic. That's fine. How is immunotherapy delivered? Is it IV or oral? Thank you. So, to my knowledge, Pembro is IV anyway, isn't it?
Yeah. Immune checkpoint inhibitors are all IV. Yeah.
Yeah. Okay.
So it is systemic. Yeah.
Okay, let's see. Lots of people saying thanks for your great presentation. Now, someone is asking about children. So, in terms of other patient factors, you mentioned patients of an older age potentially not being suitable. What about pediatrics? Is their immune system developed enough for immunotherapy to be effective? That's interesting.
Yeah, and in terms of immunotherapy for pediatrics, it tends to be hematological. There is a lot of immunotherapies approved for pediatric hematological malignancies. Generally, their immune system will have, and the problem, I suppose, the types of immunotherapies that are approved are almost ablative immunotherapies. So they tend to knock out all B cells. So if you're talking about B cell malignancies, they tend to knock them out. So it doesn't really need a mature immune system. As opposed to, they don't really, immune checkpoint inhibitors are not approved for pediatric patients.
Yeah.
Probably
and I'm
for that
also
reason.
thinking, of course, with children, we try everything we possibly can not to irradiate them, of course, before a certain age. Anyhow, someone else is asking there, is the timing of the immunotherapy with the radiotherapy important?
Yes, hugely. Yeah.
You said after. I had written, given
Yeah,
after 40 there's better outcomes. Yeah.
and the complication as well is when you bring surgery into it, because surgery generally tend to remove draining lymph nodes, that's where your source of T cells are coming from. So that complicates things further. But based on the trials so far, the most promising outcomes have been when you give immunotherapy just after radiotherapy. And I think that window, not to leave it too long, I think immunotherapy should be started quite quickly after radiation treatment, just to benefit the alterations in the tumor microenvironment.
Yeah, so sorry, I know there's a couple of other questions, but I could follow on from that because I was thinking of this in my mind, and just because we've actually had a student project talking about using photons or protons to reduce lymphopenia in thoracic cancers. And I find that really interesting that still the timing is better after, say, full dose radiotherapy, say for non-small cell lung cancer, right? Where you probably do have a lot of regions with T cells that are being irradiated, like the aorta, even the lungs themselves, So what do you think an easy, not that there's any easy wins, of course, in any of these treatments, but do you think something that we could look at, and I think the NEO Czech ray trial obviously is starting it, is to put on fairly rigorous dose volume, from our side, dose volume constraints on these organs and on blood vessels or whatever that are going to be that have T cells in them. I mean, something even as straightforward as that, if immunotherapy is still working better after RT, surely that could give maybe an incremental benefit, do you think? Or
absolutely, yeah, absolutely. Especially as the studies have shown that the T cells are so sensitive to radiation, and particularly in the lymph nodes where they're expanding, they're proliferating rapidly. That's when they're most sensitive. And they're the cells that you need to come back into the tumor. So that the ones that are in the tumor have stopped proliferating. So they're not as sensitive to radiation. Obviously, they are still sensitive, but not as sensitive. So if you could spare the lymphatics, well, maybe not all the lymphatics, but the lymphoid organs, as much as possible, I definitely think that would improve.
Yeah. So I mean,
probably incrementally as well, because there's so much going on, but I think it's a step in the right direction.
Yeah, that's interesting, because I think just for this audience, that's something we need to be thinking about then, of lymphoid tissue as an organ at risk, which we don't normally think of ever. So that's Anyway, sorry, I can come back to my questions. Let's see what else we have here. Is lymphocyte count a good surrogate for immunity of our patients? Often that is rather low in the month following a chemo orchi in stage 3 non-small cell lung cancer. I think it's coming back to the timing that you were saying about maybe start the immunotherapy not too long after the radiotherapy. Yeah. And I think the point here is that lymphocyte count is usually down then at that stage. So is that a good time to start, is what I'm interpreting. But
yeah,
who asked this can correct me in the chat or in the Q&A if I'm wrong.
Yeah, I think lymphocyte count, again, from an immunologist viewpoint, is very vague. You know, there's some good T cells, there's some good bad T cells. So an immunologist would like to be a bit more specific, and again, in circulation, obviously you're going to have lymph depletion as a result of radiation, but what's going on at the tumor draining lymph node in the tumor is most important.
Okay. Someone has written in there, some evidence that keep estimating dose to the immune cells estrogen 4.1 gray is beneficial. That's interesting, because I see that they have in that NEO check ray trial that you presented, they were looking at a lot lower than that, less than 1 gray. It's going to be very demanding, you know, with traditional techniques. But then I know you had mentioned SABR, which obviously would have much tighter margins, so that could help achieve that. And then, of course, it might help our argument for bringing protons into this country as well. So I think we'll just take one last question and then our time is up. Yeah, so there's a question there about policy levels and about the cost of immunotherapy. What actions have been implemented to make immunotherapy affordable and accessible to eligible patients? Well, that really depends on the reimbursement structure of the country, because I know we have people from different countries here today.
It is a great question, and that's something that I always bring up, health economics. It is, and Ireland are at least 2 years behind. Once the EMA approves it, it's at least 2 years before patients here can actually access those immunotherapies due to the MCP. But it is a huge factor in slowing down, you know, within this country anyway.
Yeah.
And that's why I suppose clinical trials are so important, to get patients access to these potential treatments.
Also, I think and it's really country dependent on what level of evidence is required for the reimbursement system of the country to actually say, yes, we will reimburse it. That's very different. Even in radiotherapy, that's very different from one country to another. So I think that's a huge piece of work that needs to be done. Okay, last question, and we'll let you go. How does hypoxia affect the effectiveness of immunotherapy, both in terms of effective delivery of the agent to the tumor site and the effectiveness of the agent once it reaches the target? Is it the same as for chemo? Chemo not reaching and chemo needing oxygen to be chemically active. So does it work in the same mechanism? So you had said it, I'm trying to remember what you have I learned.
No, hypoxia does itself negatively impact an anti-tumor immune response, both innate and adaptive. Both focusing directly targeting the immune cells, but also the interaction between cancer cells and immune cells. Obviously, immunotherapy, well, immune checkpoint inhibitors particularly are delivered IV. So you do need somewhat intact vasculature. But a lot of the work that's going on, I suppose, well, especially immune checkpoint inhibitors, you know, is at the tumor site. It's not in the lymph nodes. Immune checkpoint inhibitors need to act within the tumor on already activated T cells. So it is a big consideration. So hypoxia will have a negative impact as well on the success rate of immune checkpoint inhibitors in particular.
So it makes me think that, you know, even if I had my magic wand and we could all have carbon-9 centers, but like, you know, particle types that, you know, are known to be much more beneficial in radiotherapy in hypoxic tumors like carbon-9s, because, you know, of increased double-strand breaks, even in the face of hypoxia, combined with immunotherapy could be very powerful, say in, you know, radio-resistant tumors like pancreas, or, you know, something like that. So, look, we could talk about it all day, but unfortunately our time is up. Thanks a million, Joanne. That was absolutely fabulous. Really appreciate you giving your time. And I know someone has asked, can they have a copy of the presentation? So the answer to that is no, you can't have a copy of the presentation, but this recording is, this webinar has been recorded and will be on our website within the next week. So if you just look out for that, tcd.ie/radiation therapy, we will have it there in our CPD area where all our webinars are located. So if you want to share this with your colleagues, which I'm sure you do, that's where it will be. But otherwise, thanks again, Joanne. Thanks everyone for attending, and I hope you have a lovely afternoon. Bye-bye.